My name is Sofia Farina, and I'm here to talk about astrocytes and Alzheimer's disease. In the year 2050, the number of people that will be affected by Alzheimer's disease is going to be triple than 2015. So it is a urgent matter to discover more about such disease. This is how your brain looks like if you suffer from a severe state of Alzheimer's disease. And the main symptom is dementia, loss of memory, and difficulties in learning. However, what makes your brain look like this is the death of neurons. For many years, scientists focused their research only on neurons. However, it's not only about them. There are many other cells in our brain that are involved. In fact, our brain is a very complex ecosystem, and each cell has an important task for our own brain well-being. Astrocytes, in particular, they have many different tasks, one of which is to be a metabolic mediator between the neurons and the blood vessel. So let me explain better. Well, in the same way that we eat food and digest it, and this gives us energy, well, cells do the same. Astrocytes, in particular, they uptake their food, their nutrients, from the blood vessels. And they use it for their own metabolism to produce energy and other substances necessary for their life. Some of these substances are then transported to neurons, and neurons use it for their own metabolism. So you see the relationship between neurons and astrocytes is very close. In fact, in case of Alzheimer's disease, the metabolism of astrocytes can be dysfunctional, so they cannot support neurons properly. So the question is, what is the role of astrocyte in Alzheimer's disease? This is a human astrocyte in a healthy brain. And this is a human astrocyte affected by Alzheimer's disease. I guess it's clear to all the audience that these two cells have very different shape. In fact, astrocytes respond to the injuries, such as Alzheimer's disease, with changes in their morphologies. But what is the role of the change in morphologies is still not yet known. So the question that I'm addressing in my research is, what is the role of astrocyte in Alzheimer's disease? And in particular, what is the relationship between metabolic dysfunction and morphologies? So to answer this question, we use mathematical and computational tools. First, we simplified the reality with a simplified model of energy metabolism that represents the entrance of substances inside the cell, the chemical reaction, and then the export to the neurons. We then translate this into a set of mathematical equations. Then we extrapolated the shape of the real human cells and used them as domain for our simulations. So the result of my research is a three-dimensional spatio-temporal metabolic model. In particular, here you can see the healthy astrocyte and the Alzheimer's disease astrocyte. And what you see is the energy of the two cells. What we can notice is that the healthy astrocyte is much more energized. In fact, has high value of energy. However, the astrocyte affected by Alzheimer's disease is struggling and doesn't have enough energy to support itself. So, to conclude my talk, we obtain the first three-dimensional metabolic model that includes real human astrocyte, which is quite important because it's not easy to obtain human samples to study Alzheimer's disease. So we really hope that in the future, our model can be used to guide the experiments of biologists for their research. Thank you for your attention.
Thank you so much.